attended this meeting and not the other ones, and now I won't get that info. Oh, fucking sucks. I don't want to launch. Here.
Hello, the audio isn't on in virtual.
We're not able to hear anything on the Zoom. The audio is not on the virtual.
All right, testing, 1, 2.
We can hear you now, thank you.
Wonderful. All right, if I wasn't already nervous enough, good afternoon. And thank you for the introduction. While I'm before you all briefly this afternoon, it's my honor to highlight some of the initiatives and successes that we've had regarding BRS's international outreach and capacity building as it relates to engagement. As Bernadette mentioned earlier, in order to transform agricultural biotechnology regulation and to safely deliver tomorrow's innovation, we have to engage with tomorrow's innovators today. And I guess I might get to turn my own slides. Let's make sure we're going in the right direction. There we go. Fantastic. And so we did just that in FY24. We engaged with a broad array of future innovators, whether or not they be future developers, regulators, policymakers, reviewers, and communicators. And some of the examples that I wanted to share with you today are on the screen, but I really just wanted to point out. With the University of Brussels, we provided a presentation really engaged in the discussion with students in a course where the students came from over 7 countries and their backgrounds ranged from scientists to science advisors. This is something that we do on an annual basis, but we're not quite sure if you all knew about that, so we wanted to share, and one of the images here are of those students in the field. Secondarily, we also collaborate with USDA's Foreign Agricultural Service. And at that point, we work to enhance the regulatory knowledge and skills for Cochrane Fellows. And this prior year, we had the opportunity to engage with 2 sets of Cochrane Fellows, one from South America and also from Africa. And then finally, we also continue to partner with Michigan State University for their short course on biotechnology. And this really is to help provide regulatory perspectives and information as it relates to emerging issues in biotechnology regulation. And one of the things that we can say is that the future of biotechnology innovation and regulation is bright. But we also took away from that some of the areas of concern from the students and those individuals that we were able to engage. And some of those are in regards to international harmonization, as well as risk assessment, and lastly, as it relates to the application of biotechnology, in particular, how it may address both food security and climate change. Next, I said next. So BRS also continues to engage on a bilateral basis, and we do this with foreign governments as well as international partners.
And the reason why we do this is multifold. Part of this is to, again, promote harmonization, but really also to increase the information exchange opportunities that we have, but also to simultaneously bolster the credibility in USDA's biotechnology regulations while also supporting trade. And so what we tend to find is that the groups of countries that we engage on a bilateral basis may fall into two categories. And you can see amongst the countries that we have listed here, that we tend to engage with countries that may have established regulatory frameworks. And our focus really is there to not only share experience, but also to look at ways that we continue to promote harmonization. But also with the countries that are emerging or new to establishing their own regulatory frameworks, we really want to look and help and share our lessons learned to help them navigate them before they have to address them themselves. So it's better to learn from other folks, not mistakes, but their opportunities for you to enhance the way that you develop your regulations. One of the things that's not on the slide here is how many presentations and how many seminars. We had about 15 presentations and seminars, as well as 12 bilateral engagements, and that really covered the approximately 40 countries that we engage through bilateral formats and similar venues. And lastly, we have our multilateral and organizational engagements. We have and I won't mention all of the ones here, but this year I wanted to highlight that in the Trilateral Technical Working Group, the USG, which is comprised of, our delegation is comprised of USDA APHIS, FDA, and EPA, we hosted this year's annual meeting. And APHIS BRS does lead that delegation, but we were happy to not only facilitate the annual meeting, but also a quarterly meeting. And some of the topics that we discussed and focused on from a technical perspective, focused around modified microbes, which you have heard about and we'll hear about again later, as well as the ways that biotechnology can be used to address climate change and how we may be able to regulate those technologies if appropriate in the future. And as it relates to the OECD, which is the organization, because I'm not sure if everyone knows all the acronyms, but the Organization for Economic Cooperation and Development, we have a longstanding history of participation and also contributions in that fora. And this year we helped to move along and to have progress in a lot of different draft documents. But there was a council recommendations document that was revised, and this document was very important as it serves as a tool that other countries can use to establish their national regulatory frameworks or their regulations. And also, as it pertains to the Cartagena Protocol on biodiversity, although the U.S. is not a signatory, BRS does continue to collaborate with and to provide technical support to the Department of State and FAS as it relates to activities within the CBD, which is the Conventional Biodiversity. This year we did have a OTAG, and we had an SME who served as a technical expert in regards to risk assessment and development of tools regarding problem formulation, and provided support, even though they did not attend the COPMOP, did provide support as the Department of State and FAS did have personnel that were present. So I haven't been able to cover all of the activities that we've done clearly, but we did want to share some pictures and images of the broad reach that we have in the different economies and countries that we were able to engage throughout this fiscal year. So what is next for us? We definitely want to continue to provide technical support to the WTO and as well to USTR as it relates to the ongoing dispute with Mexico. And so this previous year, we did provide 2 subject matter experts from our staff that supported the Office of General Counsel as well as USTR in navigating some of those technical documents. I don't want to read all of this verbatim, but we also want to contribute to the development of a document in APEC, which is the Asian Pacific Cooperation economic cooperation organization. And then we also want to look at opportunities to engage on the regulation of novel products and to not only see how we may proceed in our own regulatory framework, but to see how other countries are approaching it. And again, harmonization is definitely always a goal, but we also want to look at how we can develop complementary approaches and to potentially navigate issues together, as we know that our legal frameworks and regulations are often different, but our scientific underpinnings and policy objectives can be similar. So with that, that does bring my presentation to a close. I'm sorry, I don't have as much presentation as everyone else does as far as our scientific reviews, but I do want to make sure that you all have an opportunity to speak with my staff if you would like to and ask any questions at this time. So thank you.
Thanks, Tessa. Hey, listen, it's always really great to hear about our partnerships and the work we do in this area.
Absolutely.
Appreciate you. I'm going to take a couple of questions for Tessa. First, I'm going to start with an online question if there is one.
Yes, it's a lengthy one. What are the key objectives for the EU engagement since EU is the main export market? Any efforts to advocate them to focus on the end-use products for safety evaluation and remove or reduce data requirements for field trials?
Well, actually, we're looking to engage with the EU and have actually spoken with individual countries that are members of the EU. I think that it's important for us to be able to engage on both technical and policy perspectives as it relates to how we regulate. And we know that with the new NGT decision that there are many things that we may know of, but that we may want to discuss on a bilateral basis. And so we're looking for opportunities to do so. And our goal is really to open the doors for communication to see what opportunities we can leverage from there. Thank you.
All right, I'm going to make a slight change in our Q&A that I should have instated from the beginning. But when you ask a question, if before your question you can state your name and the organization you're with, we would love that. I apologize that we haven't done that this far. That's on me. All right, I'm going to take a question in the room now. I see at the back of the room.
Hi, Chessa. It's Fanley Chow from the American Sea Trade Association. Good to see you. Look, I think this is a two-part question. So on the classic GMO, I think APHIS is quite progressive in its approach. So in your engagement with the governments overseas, I guess what's the end goal for that engagement? What's your message now? On the NGT or the genome editing piece, many countries, not the EU, are a lot more progressive. The EU is still proposing, their proposal is still slightly more progressive than where we're going. So what is your goal in that engagement and what is your messaging in that engagement?
Well, I see Bernadette.
Bernadette, let her handle this.
Look. Well, look, I won't phone a friend per se, but I will bring in a partner in
Let's stand here together and do it.
Yes, we will.
So on the first one, on the GMOs, when we're having bilateral engagements, we really encouraged other countries to not throw the baby out with the bathwater. Everyone is sort of getting behind this genome editing, and we want to remind folks that there are a lot of promising traits that have been and will be developed using genetic engineering that involves transgenic modifications. In that way, we also work to advocate for a more focused review, like the one that we've developed using problem formulation. And, you know, I've heard through the grapevine that we might have someone else who wants to try to use problem formulation. I'm not talking about Paraguay and Uruguay. I'm talking about someone here in the United States. And we're really excited to see more folks thinking about focusing on plausible pathways to risk and asking for those data packages more narrowly. And I think one of the things that we're able to share is how we've been able to really still address our protection goals without having these giant data packages that weigh everyone down, the developer and the regulator. So that's the message on that part.
So Bernadette, that's great. Because from a U.S. government perspective, we still have $6 billion of trade in GMOs, right? That needs to be protected. But I think one of the most progressive thing you all did was this PTMOA for transgenics, right? So how to get that message across would solve a lot of our trade issues, right? For asynchronous approvals and LLP and all of that. So I'm just kind of curious, like what's the messaging on that? What's the push on that? What's the reception on that?
So we indicate that once we've reviewed a product, we don't need to review it again, and that we've reviewed, after having reviewed product after product for 30 years, we learned that that's simply not necessary, that the risk profile doesn't change. The other day, I was saddened to know that we've reviewed glufosinate 51 times in a variety of different plants, but many times the same plant. And a 50-second review isn't going to change our thoughts on glufosinate. And I think having those types of demonstrable examples of the fact that, reviewing the same thing over and over again isn't really productive, coupled with our ability to better present and articulate the PTMOA table. If I'm being honest, what the feedback that we've received is that some people don't fully understand what we're trying to communicate through that tool. And so one of our projects for this year for Chessa's group, and I heard Chessa say she wants to spend more time up here. I was thinking, what kind of BPI can we suggest? This is a good one. So next year, we'll have a
And I will just say that I think part of the focus that we also have is communicating our continual improvement, right? And so as we think about the specific questions that you've asked, family, we definitely will hone in on communicating the things that we have domestically, as Bernadette has reiterated. So I think between the two of us, we've tackled that pretty well, but hopefully we'll be able to address that either through a BPI or some other demonstrative output.
And then regarding genome editing, real quick, because I know we're short on time and we've got to catch up. Phil, you're going to have to put the accelerator down on that top. Okay, so we recognize that what the EU's posture is slightly more aggressive than what we've just issued in our notice. We also recognize that their approach to setting their number is different. We are looking at literature that demonstrates what's achievable through conventional breeding. They've elected to impanel scientists who make that determination. Both are respectable methods of doing it. We're not suggesting that the EU needs to adopt our method of determining the number of modifications. We simply are prepared to share with them how we approached it. It's not the only way to approach it. It may not even be the best way to approach it, but it is the one that currently exists in Part 340, and that's what we follow. Yeah.
All right. Thank you. I don't have more time for questions for this session. I appreciate the interest in our international collaborations and the work we do in this area. I do need to move to introduce my next speaker. I'm going to change microphones to do that. Our next speaker is Phil Mason. He is the branch chief of our Western Compliance Assurance branch. Phil's going to talk to us about some pain points that were identified during our regulatory operations, business process improvement, and the results of that business process improvement analysis, among other things. Phil's going to talk to us about. Phil, I'll give the microphone over to you now. Thank you.
Thanks for that introduction. Nice to see you all. Well, I guess where I'd like to start today is to talk about the business process improvement that ROP, or the Regulatory Operations Program, undertook in FY24, and then quickly follow up with a little bit about the guide to reports and notices. So with that, the main goal and objective of the Business Process Improvement Project was to modernize the inspection, compliance, and enforcement processes, and really modernize and kind of make sure that we really improved our efficiency and our timeliness as we went along with this process to try to get it so that we can issue 95% of our notices of noncompliance within 14 days or less. So as we looked at the process in FY23, like Bernadette said earlier, we saw that we were issuing notices of noncompliance in FY23 in 48 days, which was really the driving force behind this project, to get us to improve those times and get things in your hands faster so that you could become compliant quicker if you had an issue. As we look to optimize our BRS resources, we looked at previous year's data, and we found that 2/3 of our resources were being dedicated towards inspections of lower risk trials. And so in order to really concentrate on trials of higher risk, we needed to refresh our risk-based inspection selection model. And as we did that, you'll see on the map on the left that really didn't have an effect on our coverage of the United States. We still had inspections from the Hawaii in the west all the way to Puerto Rico in the east, when the majority of our inspections occurring in the center part of the country. We did conduct 542 inspections last year, and we had an 86% compliance rate. So one of the benefits that we saw as a result of updating and refreshing our inspection selection model is we had more available resources because we weren't going to some of these sites multiple times under separate permits. So we had more resources to work with higher inspections of higher risk. And then one of the challenges we had was the complex review process that was causing delays in notifying our permittees of notices of non-compliance. And as we looked at time and process data for some of these processes, we found that we really could eliminate some of the handoffs, and we could eliminate some of the tasks involved. And we also found that just improving our communication between our teams and having more regular contact with our inspection and our compliance units really helped speed things up. And for instance, when we looked at an inspection that resulted in a clearly compliant inspection, we found that some of the things we changed as a result of our analyzing our time and process data, we were able to eliminate 2 handoffs and 9 tasks in the process, which really got that notice of compliance out to permittees a lot faster. And when we looked at the process in FY23, we saw that that process took us approximately 35 days. And after making some of the changes in FY24, we had reduced that to 4 days. So that was a huge improvement. And then when we looked at just handing things off from our inspection team to our compliance team, we found that we were able to reduce a few handoffs there, and that really sped things up and got notices out a lot quicker. Some other things that we did to improve some of our efficiency was we updated some of our worksheets and some of our ways that we really standardized our templates for data collection. We also updated some of our data request forms that we sent out to permit holders prior to an inspection, so that things were all set so that we aren't asking for different information for different inspections. We tried to standardize that process. And then again, another challenge we had was to develop tools to really track time and process. And one of the first things we did was the online permitting process that really, the interface that really auto-populated and tracked metric data. And then we built some different dashboards that really track time and process and scheduling of inspections and completion of inspections and things like that. When you look at the top bubble there on the right, you can see that we're able to track actually employee workloads that lets us see how many are in the queue for an inspector. Lets us see how quickly an inspector is completing those inspections, how many they've completed, how many remain, how many were compliant, how many were non-compliant. And then in the bubble below, you can see that we can track an inspection almost from the time that we issue it to the time it's completed. And we can tell how long that's taking the process for the inspection unit and how long it's taking on the compliance side if it's non-compliant. And so anywhere in the process using these different dashboards, we can see things in real time. So if there's trouble with an inspection sitting too long, if we have somebody on vacation or somebody gets sick, we're able to see that. We're able to reassign work so that things aren't sitting there. And with the inspections, we can track it through the process and find out if, you know, is the communications that we've been using to inform our compliance team that there's something there waiting for them to keep things moving, so that we can really pay close attention. And so in the future, and with this, it's really good because we're looking at real-time data and we can keep things moving, which means if there's a problem, we can get it in your hands quicker and you can become compliant quicker, and we can protect American agriculture that way. Another challenge we had was to develop a strategy to quickly assess non-compliant trends. And when we look at having the ability to look at trends, that's something we couldn't do prior to this until after the fact. And because like if we had an inspection and it was non-compliant, if we put it into something like a category that would have been records, we really couldn't do any analysis on that and find out. So we really needed to broaden some of the categories so we could break that out to be volunteer monitoring issue or training issue or things like that, or equipment cleaning. So as we broadened the categories, we also needed to train our teams on how to assign things to different categories so that we could actually do an analysis on these throughout the year. And this is really going to help us with our ability to, again, see if there's problems with the permit holders understanding the regulations, or maybe they're having problems, you know, implementing things that we can give back, and our compliance team can really work with you on some of those issues. And so that ultimately the analysis that's developed and this trends are developed into reports, and we can do automated analysis and really correct things as we go along. And then another challenge we had was to reduce the reoccurrence of non-compliance in regulated fields. And this also is associated with trend analysis, in that our compliance team can really look at some of those as they develop their strategy for progressive enforcement. They really wanted to reduce the repeated occurrence of non-compliance in regulated trials. And the way that they are addressing this is to look at some of that trend analysis, and if they can see that we're having a problem with equipment cleaning in the field, we're seeing that with one permit holder or various permit holders, they can reach out to those individuals and they can provide information for us to remain, here's some ideas to remain compliant, to get back into compliance, and to help with this not occurring again. And as we see in FY23, there was approximately 14 interactions that were proactive engagements with permit holders. And in FY24, after some of our changes, we nearly doubled and we're up to 25. So we're hoping that that'll be a trend that we will continue to increase as we go forward in FY25. And then finally, we enhanced our cross-collaboration with the APHIS investigative enforcement services, and really identified areas where they could assist us and under what areas and how they could assist us when things happen. So we tightened that up as well. And then finally, like I said at the beginning, our main goal was to issue notices of non-compliance following an inspection in 14 days or less. You can see we didn't quite meet our goal. We got to 85%. But if we look at FY23, only 15% of the time were those being issued in 14 days or less. So we had a 70% increase in things going out in 14 days or less. And if we look at the average number of days to send out a notice of non-compliance in FY23, that was taking approximately 48 days, like we said before, but in FY24, after all the changes we made and different things, we saw that we're now getting those out in 9 days. So those are some huge gains for us in about half a year that we put some of this into action. I'm really proud of the ROP team for really stepping up and reducing those numbers as quickly as we did. And it leaves us a lot of room for improvement in FY25. We're going to continue to analyze some of the changes that we made and updated with our from our inspection selection protocol to some of the measures that we put into place. We're going to continue to monitor those, and we're going to continue to implement changes as we see ways to increase our timeliness and help everyone remain compliant. So we're going to continue in FY25 to move the process even further down the line, and we hope those timelines will be further reduced in the coming year.
To the meat. Get to the meat.
And then lastly, I just wanted to touch on something that's acting on your feedback, and that's the guide to submitting reports and notices. Hopefully you all had a chance to see that. I know many of you commented on some things earlier in the year, and this was recently published at the first, I believe, in beginning of November. So it's out there and ready for you to review. I just wanted to talk about a few things that we updated to remove some duplicative language or some things that we attempted to remove that are no longer in the regulations. I just wanted to touch on a few things in e-file, specifically when we talk about the annual in-season or post-termination volunteer reports. We find that the monitoring end period date is not a required field anymore, so that's good news. Then there's some things that were in the field test reports that are no longer required in the regulations, but it was difficult to actually remove them from e-file at this time because removing them actually would have caused us some rejections in some of the things we put into the system. So we didn't want to pull that out. And I know a lot of you used some of that information, would have caused errors for you all too. So for this year, we're going to default those to no, and those are being things like unexpected effects, deleterious effects, or any planting material still growing. So those will all default to no for now until we figure out a way to remove those. And then for when we have situations of no planting, the submission and the planting and release report, we're no longer going to have a unique ID required, and there'll be no release date required. So those are just a few things there.
So if it passes
and
test,
then finally,
they have to say if it already-
again, acting on your input, I just wanted to let you know that the environmental release report has now changed from the 15th day of the month following planting now to within 30 days after planting. And so for FY25, we're going to continue to see that a notice of noncompliance will still apply for any missing environmental release report. But there'll be sort of a grace period in FY25 where a notice of noncompliance will apply to any environmental release report that is not filed either within the 30 days after planting or by the 15th day of the month following planting. So there'll be that grace period. If you do use the 15th day of the month following planting, I believe you'll get a notice of compliance with comments, and they might direct you to start to begin to use the 30 days after planting. But for now, that's what it's going to look like for FY25. So that's all I had to present today. So thank you.
Any questions? Come on.
Thank you, Phil. Impressive work from our regulatory operations team. And thanks for sharing those improvements and what they mean to our stakeholders. I'll take some questions now. I'm going to start within the room now. Do I have a question in the room?
They took away my ability to chat.
I'm not seeing anyone quite yet, so I'll go online.
Unless I'm in a queue.
A question from online from Dawn Latham is how many trials were planted in this year and what % of the trials were inspected?
So they went from raise hand to now you have to write your question during the middle of the program.
Okay. Trials were planted. What's the percentage?
Only
That, I
approved
don't have that number
questions
off the
get
top of my head.
You were supposed to.
Yeah,
like they
we
did in
can,
the
I can get
beginning.
that information and
State your name, your
post it
organization,
after
and
the
then your
fact. We
question.
conducted 542 inspections. We had an 86% compliance rate. We had 76 non-compliant notices of non-compliances issued. If that helps at all, things like that. So,
Yeah.
but I don't know exactly how the number on the other thing that you're asking, but.
We will get back on that information. Thanks. Check one more time in the room. Go ahead.
Thanks very much for the presentation. Just a note of transparency that would especially be useful for first-time permittees, especially at universities and public sector researchers. If the standard permit conditions and the so-called standard supplementals, the ones that don't change from permit to permit, if those could be placed on the web someplace so that people can see those in advance. I know a number of researchers who have felt they get up to the final stage of getting the permit issued. And if they want to go forward, sometimes it's at the 11th hour, they have to submit to whatever conditions are there. So in terms of planning, I think it would be very useful if people could see those permit conditions, both the standard in the regulation and the supplemental standard, are made available so to people who are not yet applying for permits. So right now you have to actually go in through e-file and sort of start the process. But in terms of planning, I think that would be very useful. That's not a question, just a suggestion.
Thank you.
She was begging for someone from the floor.
Thank you.
Can I provide some information on that,
you
Phil,
bet.
too? I would say our permit user guide is posted online, and in the appendices, there are our supplemental permit conditions, the standard ones that don't change from permit to permit for interstate movements of plants, for interstate movement, for importation of plants. So if you check that document online, those are there for the corn and soy release. Thanks, Dave. Okay, thanks,
you.
Phil.
Thanks.
All right, thank you. So our next speaker today is Zachary Schulthouse. Zach is a biological scientist in our plants and micro branch in our biotechnology risk analysis program. Zach today is going to talk to us about a high-level overview on the comments we received on our modified microbes request for information. He'll also talk to us about other projects dealing with modified microbes. With that, I'll hand it to you, Zach.
All right. A little more. Sorry. There we go. Let me make sure. There we go. Let me go back one. Okay. Good afternoon. As you've heard a little bit today, BRS continues to see interest in work with genetically modified microorganisms. Improving the processes by which we oversee these organisms remains a priority for us, and we have several efforts underway to achieve that goal. And today I'm going to talk about 3 of these efforts that we worked on over the last year, including some updates to permitting application flexibilities for some activities with genetically modified microorganisms, an interactive tool that we developed in collaboration with other agencies in the coordinated framework for biotechnology to assist users in understanding federal regulations governing modified microorganisms, and a little more time I'll spend on the outcome of a request for information that we published this last summer, looking for input on pathways to commercialization for genetically modified microorganisms. So from experience and discussion with stakeholders, BRS has recognized that certain features of work with genetically modified microorganisms may merit some different permit application considerations. It's much easier to modify microorganisms than other organisms. There are a greater variety of tools available and a much faster turnaround time for modifying these organisms, such that it's feasible to isolate new organisms and modify them in a relatively high throughput, almost like exploratory manner. So we are seeing more modified species of microorganisms overall than other organisms. And yes, and just a larger variety of modifications. Therefore, we've gotten together and started initiating some permit application refinements for some activities for genetically modified microorganisms. One that many of you, if not most of you, are probably aware of, we are now issuing 3-year permits for the interstate movement and importation of genetically modified microorganisms. I encourage you to make use of that if it applies to you. An additional change that we've more recently discussed and implemented is specific to the interstate movement of genetically modified microorganisms between contained facilities only. And I'll note that this does not include work in greenhouses because those merit additional containment considerations. And this new application process flexibility really is in recognition of the power of new technologies for modifying microorganisms and acknowledging the potential consequences for things that might be coming into us. Now, there is an option available for submitters to submit a single permit for multiple species. Multiple species within a kingdom on one of these types of permits. So you'll still need to submit the names of the species of the organisms that you'll be sending, but they can be grouped now within a kingdom, so multiple bacteria or multiple fungi, whereas previously we always limited this to a single genus. Oh, sorry. I don't know what's going on here. Additionally, for this option, initially, a single construct submission can cover multiple species and multiple modifications originally. And then there will be additional reporting requirements throughout the lifetime of the permit, such that additional modification information that ties to the actual organisms that were moved will need to be submitted. I'm not going to go into more information on this now, but I encourage you to look at the update to our draft guide for microbial permitting, which talks about this new option as well as provides an example permit for this. It's available on our website, and it was updated there on September 10th. Additionally, we worked in collaboration with other agencies, EPA and FDA partners, to develop an interactive tool specific to federal regulations governing modified microorganisms. It is available also now on the unified website for biotechnology, which can be accessed through this QR code. I don't know if it's really, if anyone's able to actually access that from their phones in the room, but it's on a unified website for biotechnology. You can Google it as well. It's at the bottom of the page. There's a link for this interactive tool. The way it's set up, it asks users specific questions about a product or a microorganism or an activity that they want to take part in, and leads them to specific regulatory outcomes based on their answers, such as a BRS permit for interstate movement is needed. Occasionally, when a BRS permit may not apply. There's also outcomes for EPA and FDA. As much as we were able, we also linked to guidance documents and clarified some regulatory terms relevant to different agencies, and provided data requirements and links to data requirements for following different regulatory pathways, as well as direct agency contacts for additional questions. Again, we encourage everyone to use this, and we hope to improve upon it and update it. We're able to do that as feedback comes in and as we identify ways that we can make it a little more helpful for everyone. The last thing I'll talk about is a request for information that we published this last summer about modified microorganisms. This project really emerged from comments we've had, even going back to the update on the biotechnology regulations, where there's been increased desire for us to establish and develop a process to evaluate the regulatory status of microorganisms. Just gonna put it down. Based on their plant pest risk. Additionally, we were encouraged by Executive Order 14081 to identify ways to overcome barriers to commercialization for biotechnology products. And so just after the stakeholder meeting last year, we put together a team to develop a request for information specifically focused on identifying these pathways to commercialization for microbial products and obtaining stakeholder input on this. This request for information contained 6 guiding questions that we wanted respondents to answer. It was open for a 60-day commenting period, over which time we received 50 comments, just over half from individuals and the rest from different organizations, including 12 companies and 3 universities. If anyone in here or online responded, thank you very much for providing your input. You can all access the comments online right now in the Federal Register. But I'm also going to give you a little bit of a preview or a summary of what we've been able to extract from these responses so far. I'm going to go one question at a time, not cover all of them, but give you a little bit of some details on what we've seen so far. The first question asked respondents to describe new and emerging technologies associated with modified microorganisms, and comments pointed towards new tools that could edit and modify genomes, tweak expression of genes without exogenous DNA, as well as contained uses of microorganisms for synthetic biology, such as the production of biomaterials. There was interest in that. And what I'll say, uncontained or in situ microbial tools for use in the environment, such as for bioremediation or plant growth promotion. It was also mentioned by several commenters that we should be aware of new tools that could modify or be intended to modify entire microbial communities, such that the organism that's being released may be modifying other organisms in the environment. The second part of this question asked what expertise and resources may be necessary to evaluate the plant pest risk of these tools. And it was pointed out that background and knowledge of plant microbe interactions would continue to be important, but also expertise in computational and synthetic biology and genetics to understand new technologies as well as data associated with evaluating them and new permits. The second question asked for respondents to describe areas where the clarity or efficiency of regulations governing modified microorganisms could be improved. There was a lot of interest in clarifying specific terms such as plant pest risk, as well as biological control organism and microorganism. There was interest in BRS providing resources for understanding how to fulfill specific requirements, such as SOPs that could approved or example SOPs for conducting field trials. It was suggested that we look at updating permit analysis and conditions, specifically around devitalization and persistence of microorganisms after field trials, and also aligning with PPQ on permit conditions in certain cases where we are overseeing a similar organism but a modified version. And in general, it was suggested that we collaborate closely with other agencies in the coordinated framework to mitigate overlap issues. The third question was a little more science-based. It asked about plant pest risk and how we should evaluate and view plant pest risk. The first part was about modifications that could change plant pest risk. And in response to this, it was mentioned that we should consider always the product and intent of a product or a microorganism rather than the development process. Some biological features of modifications to organisms, such as the ability to remain dormant in the environment or directly harm plants, were mentioned. Horizontal gene transfer was mentioned as a potential for plant pest risk, although we were cautioned not to take a zero tolerance approach towards horizontal gene transfer. A tiered approach towards modified microorganisms based on the risk category was recommended for our evaluation of plant pest risk. And some commenters stated that a desire for a more holistic assessment on the ecological impact of modified plant pests on plants and human health, as well as the environment. The second part of this question refocused to be consider biological control organisms and how those could pose plant pest risks. Some comments stated that a direct harm to plants should be shown before a plant pest risk determination can be made, whereas other comments stated that a specific mechanism or specificity or range of action of biocontrol organisms should be known before a determination on plant pest risk should be made. It was mentioned that we should consider in these analyses whether the context in which a microorganism can be used, such that, for example, many biocontrol organisms are already present within the environment, and think about how adding a new organism or a similar organism with a modification would change that dynamic. It was also suggested that we continue to collaborate with other agencies, again, in the coordinated framework, specifically the biopesticide group within the EPA, about overseeing plant pest risk and pesticides. Question 4 was focused on organisms with multiple uses and how we can assure oversight between different agencies. It was mentioned that nearly every microorganism can have multiple impacts, and that there should be processes put into place to identify a leading federal agency to oversee them based on the purpose of their use, and that memoranda of understanding establishing these MOUs with other agencies between BRS and them could be useful in facilitating this process. And I wanted to point out, in this case, we do have continued discussions in collaboration with EPA in developing these memoranda of understandings. The fifth question asked whether APHIS should consider risk-based exemptions for certain types of modified microorganisms, as well as ask for examples of that. In response, 5 commenters did not support such exemptions because of the novelty of modified microorganisms, whereas 18 other respondents supported such exemptions and laid out some criteria for them, including familiarity or exemptions based on our knowledge of a history of use, the purpose of the microorganism, its establishment in the environment, and known changes or effects, like modifications that we understand and are familiar with. These are just examples that commenters provided. Other commenters pointed towards current exemptions for plants that BRS already has as inspiration for exemptions for modified microorganisms, as well as exemptions for certain activities and certain organisms in other agencies such as the EPA has established. Yeah. Additionally, a common theme in response to this question was that whatever decisions we make, we should be public with the communication about the plant pest, of how we're determining the plant pest risk of microorganisms. So those are the answers to some early analysis of the answers to the questions that we've done so far. I also wanted to summarize some common themes we've taken from these answers. One is that there's continued desire for more collaboration in the coordinated framework. It was mentioned by several respondents that there's that a centralized portal would be useful for submitting applications for specific products to the entire coordinated framework. It was also recommended that we increase our transparency in certain decisions, such as communicating and explaining methods for how we determine the plant pest risk of modified microorganisms, as well as providing examples of SOPs for field trials. On the topic of permits, it was recommended by some commenters that we could look towards collaborating with PPQ for conditions on permits when the modification does not change the plant pest risk of a regulated organism. And also, we were encouraged to establish a clearer pathway for scaling up field trials. And it was pointed out that this could also encourage data collection to improve and progress the field of microorganism research as a whole. And we were also recommended to look towards exemptions that already exist for plants, as well as the regulatory status review processes that we've already developed as future motivation, inspiration, and ideas for pathways to commercialization. Okay, so this is my last slide, pretty much. We are going to continue to analyze these responses. We've basically separated them out by question, and we're going to continue to analyze them. We have a team put together for that purpose, and as well as look up resources that people provided, because some respondents provided literature to support their comments. As we move forward, we're going to try to identify ways to act on comments, some of which will probably require future rulemaking, but others we hope will allow us to. We are also going to try to use the comments to enrich our discussions with within our branch, within our microbes branch in BRS, as well as with within the agency, as we collaborate with PPQ a lot on the regulation of on how we can align on the regulation of microorganisms, and with other agencies in the coordinated framework as we discuss this issue with FDA and EPA partners and international counterparts. That's all I have. Thank you.
Great, thanks, Zach. It's really exciting to hear about the progress we're making on BRS's initiatives, important initiatives around modified microbes and their regulation. I'll take some questions for Zach now. I'll start with an online question.
Apologies if I mispronounced your name. Eleni Bickle asks, how does the current microbial permitting process manage risks linked to the interstate transport of genetically modified microorganisms for research purposes?
Okay, so we have, we treat those as interstate, as standard interstate and importation movement permits with specific containment considerations and supplemental permit conditions designed to ensure containment of the shipments. Yes, and then and we also consider containment at the destinations as well to ensure that the work is being contained there. Yeah.
Great. I'll take a in-room question. Nicole.
Hi, Nicole Juba with Bio. Thanks for the update and the great progress being made on engineered microbes. I was curious if you could share anything that's being done with international outreach and alignment on regulation of GE microbes internationally.
That is a little bit out of my arena. Thanks, though. That's, yeah. I know I had that in the very last
Partner
slide.
with us, Chessa.
Hi, Nicole. As I mentioned earlier, during our TTWG meeting or our Trilateral Technical Working Group meeting, we actually did engage around technical issues as it relates to harmonization around the regulation of modified microbes. This is an area that's very ripe for not just the U.S. regulatory system, but internationally. So we're definitely continuing to have these engagements. We're definitely not shying away from the discussions as it relates to modified microbes. I think that we have to take that into consideration as we also look at the comments that we received to the RFI. Our logical next step might be an ANPR to really look at a conceptual framework that we have and see how that fits into not only our legal framework, but also internationally. So I would say stay tuned. We definitely are going to look to have conversations this year, especially as we finalize the analysis and what we can do now as opposed to what can be accomplished through rulemaking. And I think we'll have to tier those discussions appropriately based on those outcomes and analysis. You're welcome.
Great. Thanks, Ginzak. Thanks, Chessa. All right, we have a much anticipated update. I'm going to welcome to the floor Neil Hoffman, our science advisor. He's going to give us an update on our additional modifications exception notice. Welcome, Neil.
So earlier this week, we finalized the 2 additional modifications that qualify for exemption under 7 CFR 340. As Bernadette had mentioned earlier, about a year ago, we proposed 5 exemptions, and we received comments from many of you or your colleagues. Those comments were thoroughly reviewed by us and considered very carefully. So this talk, I'm just going to give you an overview of some of the things we've heard that was very influential to us, where we ended up, practical tips on how to apply the exemptions, what is required for submitting an exemption, and what to do if a permit or an RSR includes plants that now qualify for exemption. So let's start by reviewing what types of modifications currently qualify for, well, I should say currently, as of last week, currently qualify for exemption. There are three types of DNA modifications. The first is an indel. And so a single targeted modification that involves an indel, and that's described in paragraph B1 of the regulation. And so we sometimes, you'll hear us referring to the B-type modifications. These are the B-type modifications. B1 is an indel, and paragraph B2 describes for a single nucleotide substitution. And the third one, B3, is where a gene or structural variant in the gene pool is either inserted, say through transformation, or recreated using genome editing. Now, the regulations also include a provision, B4, which allows the agency to update the modifications that are achievable through conventional breeding based on science and breeding advances. And it was that B4 a provision where we proposed these additional 5 additional exemptions. And those included greater flexibility in making indels, explicitly allowing for a deletion of any size. It described how polyploids, the exemptions applied to polyploids. It also had a provision for 4 simultaneous or sequential modifications to be made. So what did we hear from stakeholders? Well, first we heard that the proposed exemptions were too complicated. Now, the complications arose because we felt that there was, let me go back, there's a fundamental principle that we used in creating these exemptions, and that was that the exemptions should be possible by conventional breeding. There are certain types of modifications, like there's loss of function when you knock out a gene, but then there's a gain of function, which is much less statistically likely. And we thought that we needed to distinguish between loss of function and gain of function modifications, because we thought that in plants such as polyploids, which have multiple genomes, to recreate alleles that had gain of function modifications across those subgenomes would be very statistically unlikely and not likely to happen by conventional breeding. And similar arguments we were thinking for distinctions between autopolyploid plants and allopolyploid plants. I'll come back to that point later. We also proposed these 4 simultaneous or sequential modifications, and an overarching comment that we got was that was just simply too limiting, that conventional breeding allows for much more than that. And a third comment that we got, overarching comment, was that these sequential modifications should not require a voluntary confirmation request, because that then is no longer voluntary. So among the comments we got, we received many publications and some very influential ones. And one very influential one made us realize that one of our assumptions that you couldn't get gain of function mutations across the subgenomes of allopolyploids was not correct. So in this paper, and it involved wheat, these investigators had isolated a single nucleotide substitution that created a gain of function mutation in one genome, and they were able to get that exact same modification in a second genome and in a third genome and recombine them. And so they were able to create wheat and allopolyploid with 3 subgenomes having all those gain of function modifications. So that was very influential for us. It indicated to us that our basic assumption that you couldn't do this by conventional breeding was wrong. And as we explored this in more detail, we came across many advances in breeding that show the wide applicability of making these kinds of plants by conventional breeding. So where did we end up? Well, final exemptions no longer made distinctions between the polyploids. They no longer made the distinctions between gain of function or loss of function modifications. And a result of this new information, we could greatly simplify our proposed exemptions so that there went from 5 just to 2. So what are those 2 modifications that now qualify for exemption? The first one we call AM1, and AM1 stands for Additional Modification 1. And it basically is like that B1 modification where it's describing indels, but allowing those indels to be made with much more flexibility. For example, it allows the use of an external template to make deletions. It does not allow an external template to make specific kinds of insertions that just can't occur by random breaking and DNA repair. And this also does not allow certain identical deletions to be made across subgenomes. For example, if you want to make an in-frame deletion in one genome, that's a fairly low probability to get that. And if you had to make that same statistically low likelihood in-frame deletion in genomes 2 and 3, from our understanding, that's not really possible by conventional breeding. And if you know otherwise, we're happy to entertain that. So that's why we do not allow that external template to be made for certain deletions across subgenomes. But other than that, some of the flexibility that is added with the AM1 is it allows more than one cut to make a deletion within a gene or even a continuous deletion of any size. We allow a gain of function to be made from natural repair, but again, it's without the use of a template. And then there are certain cases where you can get multiple edits and it's only counted as one modification. So we base this on functionality. So if you make a change that does not change the function. So for example, there are a number of ways to make a codon, and you may make a DNA edit that does not affect the codon. So it's not a functional change. We call it a silent mutation, and those are not counted as a modification. Or let's suppose you make a knockout mutation, and that knockout, there are additional edits that happen that don't change the phenotype from the knockout. I mean, once that gene is dead, it's dead. You make 2 changes that make it dead, it's still dead. So those count as one modification. And just to emphasize, these new modifications still do not allow foreign DNA in the final product. Okay, let's move on to AM2. AM2 now says that 12 B-type modifications, and again, the B types are the indels, the single nucleotide substitutions, or the addition of an allele or a reconstruction of allele. Those 3 types of modifications can be made simultaneously or sequential. So we went from 4 to 12. And in this figure here, this picture that's showing the green chromosome, the white bands represent genes. And one point we want to illustrate is that you're allowed to make only one modification per gene in most cases. So I'm going to explain there's some cases where you can do more than one, but for the most part, you're only allowed to make one modification per gene. One question we received during the comment period, well, does this apply to plants that went through the petition process or the RSR process or the confirmation process? And the answer is yes. This applies to any plant that is not subject to 7 CFR 340. Okay, so another change we made is that the confirmation requests, we want to emphasize that the confirmation request process remains voluntary. And the change that we made, though, was that no we are no longer considering hypothetical plans for the confirmation request. Something that, when we've observed is we've had to do additional resubmissions because in the hypothetical request didn't have, when they actually made the plan, it didn't have the intended phenotype, or it didn't have the intended genotype, or the plants were not viable. So we want to reduce that number. And we also want to make sure that the plants that come in for the confirmation requests have been made. They've been planted, grown, and observed. So that was a lot of information. Now we're going to say it again, but I'm going to show you some pictures. And I will also preface my comments by saying we have a confirmation request guide, and these next series of slides are in that, and you'll be able to study that at your leisure, because I'm still going to go fast now. But I want to just touch on some salient points. So here's a cartoon that I'm going to be using over and over again. It's a hypothetical plant. We're talking about a plant that has 3 chromosomes. This is a diploid. And I want to explain how these exemptions apply to the various levels of ploidy. So diploids and various plants with greater, the polyploids. So a diploid, there are 2 copies of each chromosome. And so in this case, the chromosome, we have a blue, a green, and a purple. And notice 2 copies of each. So those chromosomes that are the same shade are considered what's known as homologous chromosomes. So how do you get polyploids from a diploid? There are multiple ways. One way is like you can take, let's look at diploid species number 1. In diploid species number 1, we could double the chromosomes. And so instead of having 2 copies of each chromosome, you've got 4. And all those chromosomes in sets of 4 are the same shade. Those are all homologous chromosomes. That would be representative of what's known as an autotetraploid. An example of an autotetraploid is potato. It's got, instead of 1 set of 3 pairs of homologous chromosomes, it's got 2 sets. There are other ways of getting polyploid plants. You can get polyploid plants by interspecific hybridization. So let's look at, we have a diploid species 1, and that could be cabbage, and that's Brassica oleracea. We could have diploid species number 2, maple leaf, that's turnip. That's Brassica rapa. Those 2 plants are different species, but they can hybridize and produce fertile offspring. And if those fertile offspring have all the chromosome sets from diploid species 1 and 2. And you can see that now we've got what we will call 2 subgenomes of 3 pairs of homologous chromosomes. You notice for each genome, you had 1 set of 3 pairs of homologous chromosomes that are all the same shade. And those, you also have one that has a different shade, but the same color, like a dark blue and a light blue. And we will call those homologous chromosomes. Because in the, say, the cabbage and the turnip, those chromosomes evolved independently because they're different species. Okay, let's look at even a slightly more complicated cartoon. This is an allopolyploid that has 4 genomes, and those subgenomes are labeled A, B, C, and D. And again, you can see in the column that's A, we've got 3 pairs of homologous chromosomes. But if you go across chromosome 1, you go from dark blue to lighter blue to lighter blue to even lighter blue. Those are the homologous chromosomes. Okay, so that's the context for now, the pictures I'm going to show you about how the exemptions apply to polyploids. So B1, that's a targeted DNA break with no repair template. This is from the old regulation from 2020. An indel modification, it only allows a single cut, no external template, and 1 pair of homologous chromosomes. And we represent pictorially various situations. So you have all the way on the left is corn with the diploid, and then you have banana. Notice that instead of 2 copies of each chromosome, we've got 3. And now we have potato, which is an example of an autotetraploid, and canola, which is an example of an allopolyploid. Now we've got 4 copies of each chromosome. And strawberry, we're saying we've got 8 copies of each chromosome. But look, we have the modification. How do we count these? One modification is if one indel, which is represented by an asterisk, on one or both pairs of alleles. So if you go at the corn, you see that asterisk is on only the blue chromosomes, and there's 1 pair, and that counts as 1 modification. Now we go to the banana, same deal. One of those copies is not allowed to have a modification. And likewise, across the board. So now you're familiar with that pattern, and I'll go quickly here. B2 now is allowing a single nucleotide substitution. 1 modification is 1 single nucleotide substitution on 1 or both pairs of alleles. Pattern looks just like the last one. So I'm going to go quickly. B3, we're introducing a gene or a structural variant from the gene pool. And that could be done by transformation, or it could be done by genome editing. But guess what? This is a turned arrow is our symbol. It's the same pattern. So I don't have to say any more about that. Now let's talk about AM1. So one point I'd like to make about AM1 is AM1 has all the functionality of B1. So you probably don't even need B1 anymore. I think B1 is now obsolete. But AM1 allows for greater flexibility. And we talked about that flexibility. You could use an external template to make deletions. You can use multiple guide RNAs to make multiple cuts to make a deletion of any size. And we're indicating that for an AM1, a deletion of any size to have a triangle. So some of these symbols have triangles. But basically you have the same pattern. 1 modification is on 1 or both alleles, a pair of alleles. So that really hasn't changed. And now the next slide, now we see a change because you can use AM2 in combination with any of those B modifications. So look at the legend all the way on the left. We have AM1, asterisk, indel, AM1, triangle, deletion of any size. We have the B2 single nucleotide substitution, or the B3 insertion of an allele or allele replacement. You can have up to 12 of those where you combine AM2 with AM1. So in this case, we've got corn, and if we look kind of underneath the corn picture, there is, we've kind of labeled what is this exemption? It's AM2 colon, and you count up how many of these other 3 modifications. This one has 8 AM1 modifications, 2 B2 modifications, and 2 B3 modifications. And you can study this picture in the guide because I don't think I have time to go over it. But a point I'd like to make is that when we're counting a modification, it's delimited by a red box. And when you've got 1 or you've got a pair of alleles modified, those are surrounded by a red box, and it's counted. So you see there are 12 numbers there. Those are the 12 modifications. Sometimes if it's a pair, look at number 8. Number 8, we've only modified 1 of the alleles. That still counts as a modification. Okay, that's a diploid. Here's the triploid. Now let's suppose we have a locus at position 1. We want to modify all 3 alleles, all copies in banana. That will count as 2 modifications because in every case, a modification is counted whether it's 1 or 2 alleles that are modified. And you can go through this at your leisure, but we have some samples, and here look at number 7. We've only modified 2 of the alleles. Number 8, we've only modified 2 of the alleles. So It's up to you. You can do whatever you want, but this is how we're going to count them. Okay, let's look at the example in a tetraploid. Let's suppose we want to modify all the alleles in the autopolyploid. Look at the, say, the blue chromosomes. And again, we've used up 2 modifications. In the allopolyploid, it would be the same thing, except we've modified 2 in the dark blue and 2 in the light blue. Let's say those are the same locus. So you're going to use up 2 modifications to modify all of those. You don't have to, you may have examples where you don't want to modify all the alleles in a given locus. Look at position number 9. We haven't modified 2 of the alleles in position number 9. And if you don't need to do that, you can increase the number of loci that can be modified. In a tetraploid, according to our scheme, if you modified all the alleles, you would be able to touch 6 loci. In this particular case, because we didn't modify all the alleles, we're actually touching 7 loci. So it's just something you can keep in mind. Last, this is the octoploid. If you want to modify all the alleles of an octoploid, and look at that first case, you're going to use up 4 of your modifications. The other point I want to make about this slide is look at the purple chromosomes. I've got triangles. I'm making believe I am knocking out the same allele on that purple chromosome in positions 8, 10, 11, and 12. I want to emphasize that you can do that if they're knockout, but you would not be allowed to do an in-frame deletion for 8, 10, 11, and 12 using an external template. Okay, you could use an external template if you were just making a knockout. Okay, now I'd like to get onto another subject. We make a distinction between edits and modifications. And there are cases where we're allowing multiple edits that only count as 1 modification. This first one I've alluded to before, where you can use 2 guide RNAs to make 2 different cuts, but whatever falls out in between, that's considered 1 modification. Here's a second example. I've also alluded to this before. 1 edit is functional, the other edits have no additional effect. So for example, let's suppose we have the sequence leucine, glycine, leucine, encoded by CTT, GGA, CTT. And now let's suppose you want to convert that glycine into an arginine. And look, you can convert that glycine into an arginine by simply changing the first G in the glycine. And just look on the right-hand side. Now it's CGA and the C is in red. That is our functional edit. We have converted a glycine into an arginine. We have made a difference. But you may make that edit and you may cause additional changes like the green A and the green G in that top one. There are 3 edits there. 1 functional change, we count that as 1 modification. Look below that. It's possible to make 6 edits there. There are 1 red and 5 green. Those 5 green do not change the sequence. We still have leucine, arginine, leucine. So 6 edits, 1 modification. And here's a third case where multiple edits count as 1 modification. And this would be applicable to that B3 modification, which, as I remind you, is for a gene or a structural variant that's in the gene pool to be added either by transformation or through genome editing. So let's look at the leaf resistance gene from wheat, leaf resistance. Yeah, leaf rust resistance gene. This is a gene that's been known for a long time. You could take that gene and you could transform it into wheat plants, and that would qualify for the B3 modification. But each time a copy of that went into the genome, that would count as one modification. Now, we also talked about recreating a gene that's in the gene pool. In cloning that leaf rust resistance gene, it was determined that it is almost identical to a gene on the 4th chromosome of the D subgenome of wheat. There are only 2 amino acid differences. Those 2 amino acids, in the sensitive allele, it's glycine 144. In the resistance allele, that glycine is an arginine. Likewise, there's a valine at the 387 position that in the resistance allele, there's a leucine 387. If you use genome editing to convert G144 to R144 and G387 to L387, you're making 2 edits, right? But that is counted as one modification for the purpose of the B3 modification. Something else I'd like to add is that, as I've mentioned many times, wheat can be a hexaploid. There are 3 subgenomes in wheat. There's a D genome, there's a subgenome, and there's a B subgenome. And there's 2 copies of a hexose transporter in each one of those genomes. There are 6 alleles. Let's suppose you changed 2 of those alleles. That would count as one modification. You just changed the 2 in the D genome. So suppose you did 2 more in the A genome. That would be 4 alleles you changed. That counts as 2 modifications. And likewise, if you did the 3rd one, that would be 6 alleles you changed, but that counts as 3 modifications. Okay, enough of that. Okay, now you know all about these new exemptions. Now you want to submit a confirmation request. Let me emphasize that this is still a voluntary process. It's completely up to you. There's some basic information we need that hasn't changed, like your name and contact information and email address, like the plant's common name. One thing that's new is now we are requesting the ploidy of the plant. This next block is very similar. We want to know what type of modification. By type, we mean, is it an AM1 modification? Is it an indel? Is it a single nucleotide substitution? Is it a deletion? Those are the sort of things we know. Also up there in that box, we say, well, there's these C exemptions. And I haven't spoken about the C exemption, but it's been mentioned today. That's where a plant has been reviewed previously. And so we did an RSR and it has the same plant trait MOA combination, or there's a petition that's been reviewed. Those are not eligible to be combined with, they're eligible for exemption, but we will not consider them if they're combined in AM2. So AM2 is only for the B-type exemptions. We also want the trait description. We want to know what gene was targeted, and then we want to know the method that you used to evaluate the plant. So there's a method that you used to make the modification. Tell us what it is. How did you verify the modification? How did you reduce modifications to similar sequences and verify that those sequences were or were not modified? And how did you verify the absence of a foreign DNA? In addition, for B3, we want to know what the donor organism was, or on what organism the modification is based, to demonstrate the modification exists in the gene pool, and that you haven't changed the expression or any of the, we say, the original genetic context. So we would expect a similar phenotype from the modification as what it was in the original genetic context. In addition, there's some helpful information that we would be happy to receive if you're good enough to send us. What is the function of the modified gene and the consequences or its phenotype. And if it has a visible phenotype, a picture of it worth 1,000 words. Molecular characterization data and sequence alignment of the modification with the unmodified sequence. Okay, that's all you need to do for a confirmation request. How do you revise permits and RSR to remove exempt plants? Okay, if you have a pending application, you can contact BRS to get rid of those exempt plants or withdraw it before the permit is acted on. What happens if the permit is issued? Again, contact BRS. You know, we've been discussing this. There are so many different scenarios that you might be in. You may not have planted. You may have planted. You may have planted, and then you may have mixed sites. So there's a lot of things that just too many combinations here to give you some advice. Please contact us and let's discuss it and how to best proceed. On RSRs, we've identified RSRs that we think are eligible for the exemptions. We will be contacting you, and we will be requesting that we set up a meeting. You can be proactive and you can request a meeting with us. So we want to discuss with you what's the best way to proceed with those. And I believe that is my last slide. Thank you.
Thank you, Neil. That did not disappoint. It was a great overview of the flexibilities provided in the recent notice, and those case studies, while complex, were super helpful. All right, I will take some questions for Neil now. I am going to start, I believe, in the room this time, and I see a hand up.
Hi, I'm Justin Shi. I just started working at USDA ARS. I have like 4 little questions. Just maybe you could rapid fire them. What are y'all doing about chromosomal rearrangements or translocations? Also,
yeah, okay. Should we just start with that one?
Sure, yeah.
Right now, we've considered that. We have decided that to leave that alone for now. So, that's something that's of interest, I know, to the field, but we have not. We do not have an exemption for chromosomal translocations.
Okay, sure, next one. What about heritable epigenetic modifications?
Heritable epigenetic modifications are not regulated.
Okay, what about using like endogenous transposons? Let's say I just want to use a retrotransposon to just copy paste like 30 of the same gene.
If there's a transposon that's in the gene pool, that would qualify for the exemption, and that's not regulated.
Okay, what about like engineered alleles? Like we can make a new allele that doesn't exist in a gene pool, but it can be made with indels or base substitutions.
That does not qualify for the exemption.
Okay, thank you.
Thanks, Justin. A question from online.
Is there an estimate of when these additional changes will be applied? And that question is from Americo Viana.
Yes, November 13th, 2 days ago, or yesterday.
I'm sorry, that's all the time we have for questions right now. I appreciate that. I will remind you that you have some pen and paper on your table. Please write down those unanswered questions, put them in the black box, middle of the room to the left side. We are going to respond to those questions and share those answers with you after the meeting. Thank you again to all our presenters. Oh, so that Neil's presentation concludes the formal part of this meeting. I'm going to invite Bernadette Warris, our Deputy Administrator, back up for some closing remarks, and then we'll have a short break. That will be followed by, at 4 p.m., an optional session by Cam von Paustuth. He is a biotechnologist in our BRAP division, and he'll tell us about some APHIS e-file updates. Bernadette, I'll give it to you for closing remarks.
Thank you. I'll make this very quick because we've gone over time. I hope you see that we left nothing on the table. We totally tried to work as hard and do as much and accomplish as much as possible over the past year. I know whatever we do never seems to be quite enough for this group, but quite frankly, we could not deliver any more than we did. I'm incredibly grateful for my team's work and effort. I'm happy that you were here today. And also, please know that we weren't able to take too many questions about the notice. Neil, you're still going to be around, walking around. Others are around here who've worked on the notice. So there'll still be time to mingle, eat food. Last year, I was worried because we ran out of food. We have more food, so eat, and then come back refreshed for the e-file discussion. Thanks.